As Jane mentioned, I'm just going to give a quick update on some sequencing that we do here at South Dakota State. So next generation sequencing at ADRDL essentially is two different tests. We run metagenomic sequencing as well as whole genome sequencing of viruses. And I'm going to go into a little bit more detail of what's the difference here and where we use these. But again, this is off our website. You can see the metagenomic sequencing, $350 per sample. Whole genome sequencing of viruses, $300. So I guess I'm going to start with the full genome sequencing, and really the question, why the full genome sequence matters. And the example I'm going to use is with PRRS, so porcine reproductive respiratory syndrome virus. I think most people in this webinar are familiar with PRRS and are familiar with PRRS sequencing. So it's very common for us to isolate PRRS or just to directly sequence PRRS. So we can compare strains. Is it a new introduction? Is it the same virus? Is that type of question. So when we do that, we sequence GP5. So this cartoon here, this diagram is an illustration of the PRRS genome. So GP5 is here. So we sequence this gene, which is about 600 base pair long. That is out of a 14,000 base pair genome. So about 5% of the genome is what we sequence, with our standard method. We take that sequence and we build these phylogenetic trees to compare strains. And then also from that sequence information, we determine the RFLP type, and this is really just an arbitrary numbering system. But it gives a very good handle, a nice handle that people like to talk about. So a 184 virus. So we talk about PRRS strains, a 184 or a 252. It's just a nice naming scheme so we can have an easy way to describe these viruses. But again, this is all determined based on the GP5 sequence, which is traditional technology that we still do at South Dakota State. That's been around for several decades now. So the problem with just sequencing GP5 is you're really only getting a small part of the story. These bullet points here kind of highlight some of the reasons why we'd want to sequence more of the virus. When we look at the structural proteins that are important for PRRS immunity, there's a number of proteins other than GP5, and these other proteins are the target of neutralizing antibodies. So again, it's important to look at, to have the big picture here and not just focus on one part of the virus. Also, pigs are frequently co-infected with multiple strains of PRRS. So as the traditional Sanger sequencing of GP5, you're only going to be able to sequence one strain of PRRS. With the newer next-gen sequencing, we can tell if a pig has multiple strains of PRRS. And this is actually fairly common. I've seen reports in the literature that probably on the neighborhood of 5% of pigs that are PRRS positive are actually infected with multiple strains of PRRS. This is important because if you have co-infections in one animal, these strains can recombine. And I'm going to show another slide on that in just a second. But again, recombination is how the virus evolves. And again, if you're only looking at a tiny part of the genome, you're not going to realize the virus is recombinant. And then finally, the pathogenicity of PRRS is linked to non-structural proteins. So when we think about a really nasty PRRS strain, a lot of those genetic changes that make PRRS nasty are in the non-structural protein region of the genome. So again, that's why we want to look at the virus as a whole. So again, I mentioned GP5. This is a cartoon of what the virus looks like. GP5 is on the surface of the virus, but you can see here there's a number of other proteins on the outside of the virion. So when the PRRS infects a pig, the immune system is seeing all these different proteins and it's generating a response to different ones. So again, it's important to look at all these different genes that encode for the different surface proteins in particular. I mentioned recombination. This is a paper that was published just recently. where researchers at ISU identified a recombinant virus between a vaccine and a wild type PRRS strain. And again, what happens here is if I've got two strains of PRRS, so let's say a virulent wild type strain here in blue, and then this Fostera vaccine strain in green, if both those viruses get in the same pig, they can recombine. And what essentially comes out is this strain up top, this recombinant PRRS. So the front half of the virus is actually the wild type field strain, but the back half looks like Fostera PRRS. So initially, when the researchers at ISU identified this strain, they were sequencing just GP5, which is down at the end of the genome, and it looked like vaccine strain. But in reality, when you look at the full genome, you see that it's not a vaccine strain. It's essentially half vaccine and half wild type, so a chimeric virus. But again, it's the kind of thing you can only determine if you're doing full genome sequencing. And then finally, this is a very ugly, complicated picture, but the point here is what I'm looking at is the non-structural protein region of the genome. And you'll see there's all these little numbers and little holes. And essentially what that means is that that particular region of the genome is very elastic and it mutates readily, and we see a lot of deletions and insertions. And what we've seen with time is as PRRS evolves, oftentimes we see increasing deletions in this region of the genome, which is often tied to increased pathogenicity. So these strains mutate and they become more virulent. But again, you need to look at the whole virus to see these changes. So again, that's kind of a quick summary why we do full genome sequencing. And really it comes down to a more comprehensive picture of the virus on a farm. And again, as we use this with time, this is very valuable. We can see how the virus is changing within a farm, how we're seeing new introductions horizontally of virus. And really the goal here is to correlate these genetic changes to changes in management and health outcomes. So that's full genome sequencing. I'm next going to talk about the metagenomic sequencing, and I'm going to start with the definition of metagenome. I think that's a term that probably not everyone's familiar with. What the metagenome is all the genetic material in a sample. So for the purpose of this talk, the sample being, say, a nasal swab or a fecal swab or tissue from a pig. So what genetic material is in that swine nasal swab? So if we think about, if I stick a Q-tip in a pig's nose, what's there? I'm going to see DNA and RNA from the pig. I'm going to see bacteria. I'm going to see bacteriophage, which are viruses that infect bacteria, as well as viruses. Now, all these bacteria and viruses are a combination of just environmental bacteria and viruses, things that, the pig's breathing in and that's getting into its nose, for example, as well as bacteria and viruses that infect the pig. So again, it's a very complex mix of environmental as well as pathogens, bacteria and viruses. So we do this metagenomic sequencing. The difference is, between like the standard sequencing and the metagenomic sequencing is the metagenomic sequencing generates just a massive amount of information because, as I explained, it's a very complicated sample. So we need to sequence a lot of different things. So we apply this for a number of different cases diagnostically. These bullet points here kind of describes generally where we use metagenomic sequencing. Probably the most common is unusual clinical presentations, and I'm going to give an example of this. But basically, where I've got pigs with clinical disease that's unusual, or when we test pigs with clinical disease and we don't identify what we suspect. We think it's PRRS, but we do a PRRS PCR and it's PRRS negative. Well, the pig is sick, but we don't know what's causing it. So really, we do the metagenomic sequencing because it's a very general way to identify everything that's in there. We also use this for herd profiling, screening feedback or exposure inoculum, and then in particular, looking at complex diseases such as respiratory disease complex, where the clinical outcome is really the interaction between the host and bacteria and viral factors, where there's a lot of different things at play that result in disease. It's not simple single pathogen leading to disease. So this video here was actually from a case that I received when I was down at K-State. And again, this is a pig with neurological disease. Neurological disease diagnostic investigations are often fairly tricky. There's a lot of different organisms involved. And a lot of these organisms we don't have standard diagnostics for because these tend to be fairly rare diseases. So this pig, the tissues originally came in for testing, tested negative for the usual suspects. This was actually a fairly big break of disease. We had 700 pigs involved, essentially with attention tremors, increased respiratory rates. And as the disease progressed over a period of 4 days, 100% mortality. The pigs essentially couldn't walk, couldn't get to the feeders, couldn't drink. Standard testing identified nothing. We did next-gen sequencing, metagenomic sequencing on the brain, and identified atypical porcine pestivirus. So this was a virus we had previously identified down at K-State. We didn't know what clinical significance it had, but in this case here, we identified it associated with neurological disease. Other researchers at ISU also working with this virus were able to show that infection in utero can cause congenital tremors. So again, clinical disease, we weren't sure what's going on, but the next-gen sequencing, metagenomic sequencing allowed us to identify this novel emerging virus. But down below are a number of viruses that we commonly find in neurological disease, including in South Dakota, saplovirus, teshovirus, astro3, and PERS. Again, a lot of these viruses we don't typically test for. The saplo, tesho, astrovirus, they're fairly rare in terms of us identifying them in diagnostic cases. So really the metagenomic sequencing is nice because we don't need to know what we're looking for. Another example, this was again from when I was at K-State. This is where we discovered a new species of Circovirus, porcine Circovirus 3. This falls under the category of cases where we think we know what we're looking for, but we don't find it. So in this case, we had sows which died with PDNS, so porcine dermatitis nephropathy syndrome, with lesions like this. And the sows also were aborting. Typically, this is associated with PCV2 infections. However, all the diagnostic testing at ISU failed to identify PCV2, so that's where we got involved. Samples were sent to K-State. We ended up identifying a novel circovirus, which we named porcine Circovirus 3. If we look at the genetics here, PCV1 and 2 right here, very distantly related to PCV3. So again, a novel circovirus, we did a bunch more work with it and showed that there were very high levels of PCV3 in these mummies. This virus is fairly common in the US swine herd. And then importantly, when we screened archive samples of cases of PDNS that tested negative for PCV2, we found that 94% of them were positive for PCV3. So this was back in, I think, 2016 when we published this work. PCV3 is becoming a bigger and bigger deal. Merck is producing some vaccines against this now, and I do know a lot of veterinarians have experienced problems due to this virus. So this certainly would fall in the emerging virus camp, where it certainly seems to be causing some problems in the field. But again, the metagenomic sequencing enabled us to identify this virus, which really set that ball in motion for the diagnostics and vaccines. Final example here, this is a more recent one from September here in South Dakota. This was a case that came in for PERS and mycoplasma testing. This was a gilt. Dr. Lynn, a pathologist, identified pneumonia via histopathology. However, so he identified monocytic interstitial pneumonia by histopathology, which tested negative for PERS, flu, and myco. So something's going on, but we weren't sure quite what it was. We performed metagenomic sequencing on those and identified porcine parvovirus 2. So this is also an emerging virus. There's been a few papers on this, but really the etiology and the significance of this virus is unknown. However, there are reports anecdotally that suggest this is involved in respiratory disease as well as potentially lameness. So Dr. Lynn and myself just recently received a grant from the Swine Health Information Center to further investigate the role of Parvo2 in clinical disease. So to conclude, both these technologies, the metagenomic sequencing and the full genome sequencing, are very powerful techniques for us to use in veterinary diagnostics. If we talk about full genome sequencing, it's a very precise way to monitor the evolution of these viruses. And then the metagenomic sequencing allows us to detect all these viruses and bacteria present in a sample without knowing what we're looking for. And that's really the value of that approach. Is I don't need to know I don't need to ask the question, is it positive for PRRS or PCV2? I can just ask the question, what's there? Pretty neat stuff. So I'd be happy to answer any questions if you have any.